Bismillah ar-Rahman Rahim, 9700 June 24, paper 4-3, and this is the first video which will discuss the first three questions. As you all know, this paper is for 2 hours and we should try and finish this well in time so that we can revise it. Question 1, figure 1.1 outlines the effect of ADH, antidiuretic hormone, on the cells of the collecting duct. The cell signaling mechanism of ADH is similar to that of glucagon on liver cells. So you have a diagram here, you can see the blood here, and you can see the hormone ADH traveling in the blood and then moving out of the capillary and getting attached to the receptor protein and then causing ATP is being converted, and then we have an enzyme cascade, and then we have these vesicles, the aquaporins, coming and attaching to the cell surface membrane. So you have A, B, C and D. Next, they want to know, name the structures A, B and C. What is A? A is a G protein. And what is B? Is cyclic AMP. And what is C? It's aquaporin. So A is a G protein, B is cyclic AMP, or you can write CAMP as well, and B is an aquaporin. And then we come on to the B part of the question. It says ADH is secreted by the posterior pituitary gland when the water potential of the blood decreases. Suggest reasons why the water pressure of the blood may decrease. Well, if you don't drink enough water, like we said, dehydration, or if you ingest too much of a salty food, if you had too much of a chicken kadai with lots of salt in it, or you've had food which has a lot of carbohydrate in it, so a lot of glucose and amino acids have entered the blood, or you have sweated a lot, you've been playing basketball and it's been a bright sunny day, so you've been sweating a lot, so there's a lot of water that has been lost. So all these factors would make the blood very concentrated. So basically you got to word it correctly: dehydration and not drinking enough water, or ingesting salty food, or if you had more ions, we had food which was rich in glucose and amino acids, and so all those would be digested and then absorbed into the blood. Then sweating or perspiration or evaporation of water. Then part C of the question says diabetes insipidus is a condition affecting osmoregulation by the kidney. One form of diabetes insipidus is caused by a tumor in the pituitary gland, which results in a decreased secretion of ADH. Suggest the symptoms that would occur in a person with diabetes insipidus. That means less ADH. Less ADH means less water reabsorbed. So increase in the urine volume, or you could have said more frequent urination. So increase in urine volume. Number 2, fatigue. Number 3, feeling of thirst. And could have said dehydration, or there could be also a change in blood pressure, because ADH is secreted when the blood becomes concentrated. So if it is not going to be secreted, then the person's blood concentration will remain very concentrated, and that will not be a very good sign, and it will affect the body tissues. So the person is going to feel tired all the time, fatigue, feeling of thirst. You see, when your blood becomes concentrated, besides ADH being produced and increasing the permeability of the collecting duct and water being reabsorbed, but now what is going to happen? The person, it says, results in decreased secretion of ADH, so the blood will remain concentrated. So there will be increase in the volume of the urine will increase, and fatigue, feeling of thirst and dehydration. So increase in urine volume, fatigue, feeling of thirst, dehydration. Now we come to the D part of the question. Neurogenic diabetes insipidus, NDI, is another form of diabetes insipidus. In NDI, ADH molecules cannot bind to the receptor protein located in the cell surface membrane of the cells of the collecting duct. With reference to figure 1.1, explain the effect on the cell surface membrane labeled P if ADH cannot bind to the receptor protein. If it cannot bind to the receptor proteins, what will happen? No aquaporins will be added to the membrane. So there will be low permeability of the membrane. Less water will move through the membrane P. So if ADH doesn't bind, then nothing's going to happen. It's not going to alter the permeability. So water will not be reabsorbed. Water basically is reabsorbed back into the blood because the blood is very concentrated and we want it to return to the set point. So no aquaporins added to membrane, low permeability of the membrane to water, less water moves through the membrane. But you were only supposed to give me 2 points because there are only 2 marks for this. NDI is caused by a recessive allele of the gene coding for the receptor protein. The gene is located on the X chromosome. Explain why a man with NDI could not have inherited this condition from his father. Wow. You see, what do you get? If the mother is XX and the dad is XY. So if this couple is going to have a son, they get the Y. If this is going to be a male, then he gets the Y from his dad. So any son to a couple, he gets the Y from his father because the mother doesn't have a Y. He only gets the X from his mother, one of the X from his mother. My mother has 2 X. So which X he gets, there could be this one, could be this one, could be this one, could be this one. But I mean, if this couple has to have a daughter, then they get the X from the dad. So actually it's the male, it's the sperm which decides the gender of the baby. So that is why the Y sperm, if that fertilizes, then it's going to be a male child. If the X sperm fertilizes, then it's going to be a female child. So explain why a man with NDI could not have inherited the condition from his father. Basically, X chromosome with the recessive allele is inherited from the mother because the gene is on the X chromosome. And the Y chromosome is inherited from the father. So that is what you had to give me as an explanation. So the simple way how you wrote that was X chromosome with recessive allele inherited from mother and the Y chromosome inherited from father, which does of course not contain the recessive allele. Question number 2. Phenotypic variation exists in many forms. Some examples of phenotypic variation in plants and animals are described in table 2.1. Complete table 2.1 by stating whether the cause of variation for each described example is likely to be due to genetic factors VG. Genetic factors, so we write VG. And then environmental factors, we write VE. And a combination, so we write VG and VE. Now, as we look at all of them, we need to read them very carefully and we need to sort of look at it and see which one is right and which one is which. Tomato plants grown in a glass house and grown outside vary in the yield of tomatoes they produce. 17 genes associated with tomato yield have been identified. So tomato plants grown in a greenhouse and grown outside vary in the yield of tomatoes they produce. 17 genes associated. So it's VG and VE. So this would be VG plus VE. Now, new strawberry plants of the variety called sweet Anne are made by asexual reproduction. The new plants grow to different sizes and produce different number of fruits. So, new strawberry plants from the variety called sweet Anne are made by asexual reproduction. The new plants grow to different sizes and produce different numbers of fruit. Now, what would this be? Why do they grow to different sizes and produce? Because it's not genetics. It's all asexual reproduction. So it means maybe somewhere the soil is more fertile, more nitrates, more magnesium. So this would be VE. And then it says, number 3, the domestic cat has a blood group system with 3 possible blood types, A, B and AB. The blood types are determined by antigens present on the cell surface membrane of red blood cells. So phenotype would be what? VG. Why VG? Because there are three possible blood types and determined by antigens present on the cell surface membrane. It's nothing to do with the environment. You might starve these cats, but they're going to remain the same blood groups. Then coming on to the next one, over 50 genes have variants that are associated with excessive weight gain in humans. Other risk factors for excessive weight gain include diet and exercise. So over 50 genes. So we're talking of genetics and then we're talking of diet and exercise as well. So this is VG plus VE. Then we come to the next one, the last one. Resting heart rate in human varies between different individuals. Some factors that influence resting heart rate include biological, sex, family, history of heart disease, number of cigarette smokes, and medication taken. This is again VG and VE. Because you see, they're talking of number one, they said biological, then they said family history, so that's genetics, then heart disease, family history of heart disease would be genetics, and number of cigarettes is against environment, and medication taken is again environment. So if you got all five correct, you got 3 marks. So that means it was a very easy question, and that's why it's not for many marks. Then it says name a spontaneous random event occurring in cell, that means source of variation, that's mutation. It's very obvious, there's no other answer for this. Then it says, other than the event named in B, describe the features of sexual reproduction that contribute to the production of genetically different offsprings. So in B, they said don't talk about mutation, but then talk about other things. Now the other things will be what? In sexual reproduction is crossing over, independent assortment, random fusion of gametes, and random mating. That's of course a chapter, natural selection and evolution. So the other causes of genetically different, why would a couple have 4 sons or 4 daughters and they're all very different? Basically it's because of crossing over, because of independent assortment, because of random fusion of gamete. Which gamete fertilizes? There are millions of sperms. Which sperm fertilizes that ovum? And random mating, of course we're talking of animals here then. So crossing over, independent assortment, random fusion of gametes, and random mating. Question number 3. In plants and humans, the phenotype of an organism is determined by the genotype and the environment. Plants from the genus Primula have different petal colors. The presence of the pigment malvidin results in blue petals. The metabolic pathway for malvidin synthesis is controlled by gene bada T, capital T, and small t. The presence of the dominant allele capital T results in blue petals. So that means the blue petals will be either be big T or big T small t. I always tell you that you need to figure this out in the beginning of the question. Another gene D, capital D and small d at a different locus also influences the malvidin synthesis pathway. When the dominant allele D is present, this gene product suppresses the malvidin synthesis. So this is summarized in figure 3.1. Precursor allele T, malvidin, and then blue petals. But then precursor allele D, no malvidin, so non-blue petals. Right? So the presence of big D, the dominant allele big D, results in the suppression or the malvidin synthesis pathway. Now a genetic cross was carried out between 2 plants heterozygous at both gene loci, and the resulting offspring genotypes are shown in a Punnett square in figure 3.2. So 2 plants heterozygous, so big T small t and big D small d at both gene loci were crossed with each other. So we have big T small t and big D small d. So they are crossed and the resulting offspring genotypes are shown in a Punnett square. State which of the genotypes shown in figure 3.2 have blue petals. That's 1 mark. That's 1 mark. So which ones will have blue petals? Basically the one which doesn't have the big D. So it will only be so basically it is going to be the it had to have the homozygous recessive. So it had to have small d in both the situations. And these would be, of course, the one which have blue petals because the big D is the suppressor. Now it says state the ratio of the non-blue to blue petals for the cross shown in figure 3.2. Non-blue 2 is 13:3. You see all the others. Now this was how many? They were 16. So if 3 of them are blue, all the others are non-blue. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this was 13:3. The ratio of non-blue to blue petals. So the blue ones, we did that earlier. These were the blue ones. These were the blue ones. So we have 3 blue ones and 13 are the non-blue ones. Then it says name the type of gene interaction that is called the offspring ratio you have stated. So that is epistasis. So this has got you your 1 mark for writing epistasis. Then part 4 of the question, it says gene capital T small t and gene capital D small d code for proteins that are involved in the control of the production of molybdenum. Discuss the possible roles of the proteins coded for by the gene TT and gene DD in the control of the production of malvidin. So dominant allele capital T and capital D results in a functional protein. Then the 2 small recessive alleles small t and small d result in a non-functional protein. Capital D code for an enzyme, and this enzyme catalyzes the production of molybdenum, which is the pigment. Capital D could code for an inhibitor. Why do we say code for an inhibitor? Because you see it is not resulting in the pigment being produced. So inhibitor binds to enzyme coded for by the gene capital T. So reaction to produce molybdenum does not occur. So reaction to produce pigment does not occur, or you could have said the protein coded by capital T could be a transcription factor. And the transcription factor allows the production of the enzyme needed in the metabolic pathway. Now, the protein coded by capital D could cause the inhibition of the transcription factor. You see, if the transcription factor is inhibited, then that protein will not be made. So there were about 10 mark scheme points, and you had to give me any 5 of those. The possible roles of the protein coded. So dominant allele results in a functional protein. 2 recessive alleles result in if the 2 of them are together, small t, and small d, result in a non-functional protein. So the capital T code for an enzyme. Enzyme catalyzes production of pigment. D could code for an inhibitor. Inhibitor binds to enzyme. So reaction to produce pigment does not occur. Protein coded by T could be a transcription factor. Transcription factor allows the production of enzyme. Protein coded by D could cause the inhibition of the transcription factor. So understand is that you see what does it mean capital D, and what does it mean if it's a dominant phenotype, and what does it mean it's a recessive phenotype. So please be clear about that. So just have a look at all this. I mean, these are all the things that I have said to you and I have repeated them to you. You can pause the video here and have a look at it. And if there's anything not clear, then you need to really revise the chapter. Then we come on to the B part of the question. Some humans have the inherited condition hemophilia. Explain the relationship between the F8 gene factor 8 and the condition hemophilia. So located on the X chromosome, it's a recessive allele, non-functioning factor 8 protein. Blood does not clot quickly enough. Excessive bleeding occurs. And then, the phenotypes, we could link the phenotypes to the genotypes, and it prevents the action of fibrinogen not being converted to fibrin. So the blood is not going to clot. So located on the X chromosome, recessive allele, non-functioning factor 8 protein, blood does not clot quickly, so the person is going to die of excessive bleeding after an injury, prevents fibrinogen to fibrin. Then the genotypes, so these are all normal females, these are normal males, this is the sufferer, and this is the female sufferer. But this female sufferer does not exist on the face of this earth because this is a lethal mutation. And if this is present in the zygote, the embryo will not develop from it. Question number 4. Genetic engineering is a technique used to modify the genetic material of a specific organism to change a characteristic. Genetic engineering uses specific enzymes and commonly involves the use of plasmids for the transfer of genes into an organism. 4 enzymes that are used in genetic engineering technique involve plasmids are restriction endonuclease, DNA ligase, DNA polymerase, and reverse transcriptase. Outline the roles of these enzymes in genetic engineering involving plasmids. So restriction enzymes we use to cut DNA or to cut plasmid. DNA ligase joins the DNA with the plasmid. Then DNA polymerase forms a double-stranded DNA, or you can say makes a complementary DNA strand. And then the last one is reverse transcriptase. Now that is used as mRNA as a template to make cDNA, which is complementary DNA, or you can say ssDNA, which is single-stranded DNA. So uses the mRNA as a template to make cDNA. cDNA stands for complementary DNA, or you can say single-stranded DNA, ssDNA. So restriction endonuclease is used to cut DNA or plasmids. DNA ligase, we use it to join the DNA because we've got the plasmid. So we've cut the plasmid, and now we've added the gene of interest. Now we needed to glue it together. So we needed to glue it together, and we need the enzyme ligase to make the phosphodiester bond between two different DNAs from different species. DNA polymerase is, you see, you've got one strand of DNA. Now you need the second strand of DNA. For that you need DNA polymerase. So from double-stranded DNA, right? Then we have the last one. It says reverse transcriptase. So you've got the mRNA. Say this is the mRNA, right? And then you're going to give it the reverse transcriptase. It's going to make one strand of DNA. This DNA does not contain the introns, so that's why it's called cDNA. The normal DNA has introns and exons. But then remember, from the DNA it becomes pre-mRNA, and then it becomes mRNA. Now you've taken the mRNA out, and you're giving it the nucleotides, and you are giving it reverse transcriptase enzyme. So it's a reverse transcription. Transcription is what? DNA to mRNA. So basically the mRNA is being used as a template strand to get cDNA. Why is it called cDNA? Why isn't it called the ordinary DNA? Because in the normal DNA in eukaryotes, there'll be introns. And if you've taken the insulin mRNA out of the beta cells of the pancreas, well, that RNA has been processed. And from that RNA, the DNA that you're going to make is going to be called cDNA. Then part 2 of the question says, explain why a promoter as well as the desired gene is often transferred into an organism. Yeah, because you see, the promoter is where the RNA polymerase binds. So for transcription factor to bind, or for RNA polymerase to bind so that the gene is expressed, or we can say the gene is switched on, and to increase the transcription of the gene of interest. I mean, I want the insulin gene to make insulin for me. So I've got to somehow make the bacteria make insulin for me. So to increase the transcription of the gene of interest and to cause transcription with a specific environmental change. So we want the promoter has the one where the RNA polymerase recognizes only the area which is the promoter area, and then it binds, and then transcription starts. So for transcription factor to bind, for RNA polymerase to bind, so gene is switched on, or you could say gene is activated to increase the transcription of gene of interest. We want the insulin gene to start working and produce more and more insulin for us. That's the main purpose of genetic engineering. Part B of the question says the production of insulin by genetic engineering involves the use of plasmids and the bacterium Escherichia coli. Multiple copies of a gene that codes for an insulin polypeptide are mixed with cut plasmids. During the process, only some of the plasmids are taken up by the host bacteria, will lead to the expression of the insulin polypeptides. A cut plasmid and the gene coding for the insulin polypeptide, figure 4.1 shows that 3 different plasmids have been formed as part of the genetic engineering process. So we look at the diagram and we say complementary sticky ends ready for gene introduction, promoter, marker gene, key, gene that codes for insulin, direction of transcription. So they've given you all these clues, and then they've given you 3 of these plasmids, plasmid X, plasmid Y, and plasmid Z. Now, and then what do they ask you in the next question? It says, comment on whether a bacteria will produce the insulin polypeptide if it has either plasmid X, Y, or Z, and explain the reason for your choice. And that's 3 marks. So let's look at it again. Plasmid X, why would it? No insulin because the genes is inserted backwards. This works like this transcription. This is backwards. So no insulin because the gene is inserted backwards, the wrong way around. In Y, yes, it will produce insulin because the gene is inserted in the correct way. Transcription goes like this. Now Z, no insulin because the gene was not inserted. There's no gene here. This was the marker gene and this was the promoter. But there is no gene here. This didn't come in here. This didn't come and get fixed here. So no insulin because the gene was not inserted. The plasmid just closed up. It closed up. It's a non-recombinant plasmid. It doesn't have the gene of interest. So basically no insulin gene inserted backwards, so no insulin will be produced. In Y produces insulin because it's correct way, and Z there's no insulin because the gene was just not even put. The plasmid just closed up without the gene. We will continue the rest of the questions in another video. Thank you very much.